Hi everyone. In this video, we will do another interesting use case on that means AI assisted single cell data analysis using MCP and Scanpy. So basically, Scanpy is a very popular tool that is used for single cell data analysis using Python. And basically, we will take this Scanpy, steps and convert it into different Python functions and package it into a MCP server, and then so that our AI assistant, like for example, Cloud Desktop, can interact with this MCP server and can help us to do single cell data analysis, right? So this is just give you some example on like how we can build an alternate MCP server that is single cell data analysis. So very simple, same process here. We will use first MCP to build the MCP server. So we will take all the Scanpy related QC functions here, and then we will run it via not Docker run actually. We'll run it directly. We'll not use Docker here. We will run it directly and in the form of HTTP mode, and so that it will be exposed in some URL. Then we will take that and we'll configure our Cloud Desktop so that it will connect to that MCP server and it will help us to do the analysis. Okay, so coming to you can go to Scanpy. Okay, Scanpy. So this is the tool that is helpful for single cell data analysis. So here what I did, I went to this tutorial and just for the basic pre-processing and right, so I followed those tutorials and just to convert that into tools. And so this is what I followed some examples over here like quality control, then just to give you some idea, so that to set the context. So basically single cell data is usually in the form of either matrix format or this H5 format. So you load that and once you load that, then you can do quality control analysis. So there is a function called quality calculate QC metrics in Scanpy, which will help us to calculate basic QC metrics. And we can also add some number of mitochondrial genes, also ribosomal genes, and hemoglobin genes as a QC parameters. Like for every cell, these parameters will be calculated by this three snippet, and then you can do calculate QC metrics, and then you can use this parameter too, so that those QC metrics will be calculated. And then using this Violin plot, here you can pass what all QC metrics you want to see. For example, it will give a Violin plot of number of UMIs, number of genes by count, total counts, percentage mitochondrial contaminations, percentage of mitochondrial genes. So those Violin plots will come. And also you can do a scatter plot. So here you can pass any two variable and you can color with other parameter to see how these two variables are correlated with each other. So this is one functionality of doing scatter plot, and here one is the Violin plot. And after that you can using this filter cell and filter genes, you can filter number of for example any cells where number of genes expressed is not 100 or less. Similarly any cells where number of genes expressed means like remove those genes which are not expressed in at least minimum 3 genes, it should be expressed in the 3 cells. So that kind of, so 2 steps, what we can remove those bad cells, and here we can filter bad genes, we can say, or low expressed genes. Optionally you can have some, for example, doublet detection. You can use scrublet to calculate the doublet score, and also normalization is there, and you can have feature selection like identify highly variable genes, and you can do even PCA dimensional reductions, and this kind of plots you can do, and calculate neighbor nearest neighbor cells, like those cells whose profiles are very similar in terms of expression, those will be clustered together, and you can draw a UMAP plot. Okay, and also you can do the clustering to get this kind of output as a latent clustering you can do. So you can see that they have given some tutorial. So what I had done, I convert that into sorry for that. I converted those functions into those features into basic Python functions. So I written a very simple mcp.py. By the way, this is in a different repository. Why? Because I want to also show you in the next video. Once you build into the MCP server, how you can share it to the other people in the form. So that you remember in the first video, we started with using others MCP server, right? Like Uniprot MCP server. So that's what I will show you, like how we can take our code and do some changes, and so that we can other people can also use it. Okay, so that's what also I'll show you that. So that's what I have created another separate repository, single cell MCP server, and within that, in src folder, single cell MCP, the Python code is there. So code is very simple. Like again, we need this fast MCP for building MCP server, and we need Scanpy for installation. We need ANN data, annotation data, and we need matplotlib, and we also need Scarlet, and those kind of basic things we need. Okay, so these are the packages we need, and here we are defining two global variable. A data is where all our annotation data will be stored, and QC summary is a variable where we are storing the QC region. So the process is very simple. See, I am creating an MCP object, that means the first MCP server object. Then I have just created a very greetings tool. This is a very simple tool which will just say that, hey, hello from single MCP. This is sometimes useful to introduce this kind of greetings tool, because that's where your MCP client can connect to and call this greet function to check whether MCP server is running properly or not. Okay, so I've defined a very first tool here. So that is what load single cell data. So here I've given the instruction that load single cell data from a file or folder. It supports both H5AD file or even 10x folder. So in a 10x folder, like we'll be having 3 files like barcode, the genes, the matrix. Okay, and this is a binary file. So those two kind of datas are there, and we need to provide the support for both. And so that's why I have written that if the folder contains H5AD files, loads first one found. Okay, so if multiple is there, then whichever is first, you take it. So very simple function. So you define a global A data, means it will point to our this global variable, and here you see I have imported the OS module, and if the path is present, and if is a file, and if it is H5AD file, then you using this function read H5AD. Let me close that, so that okay, this function you will load the binary file. Otherwise, if it is not H5AD file, it will check whether yeah. So if it is multiple files are there, it will choose the first one. Otherwise, it will try to load the 10x file using read 10x mtx function using the Python package, right? sc.read 10x, and ultimately this will store the data, A data, and it will give a response at the end that, hey, loaded the 10x folder and how many cells and how many genes it found. Okay, and if some exception comes, it will say that folder doesn't contain either this file or valid 10x file, so it will give some error, or it will give some response that there should be file or some 10x formatted file should be there. So this is a very basic function which I have converted a package that simple 2 lines what you see there into very manual function, and decorated it with under @mcp.tool. Now it become my single cell first tool. So now I have created a simple function, single cell data summary. Basically this will give me this 4 information: how many cells, how many genes, what are the gene names, top 5 gene 5 gene names, and what are the variables or the cell names. Okay, so this is the single cell data summary function. Now we have a function called calculate QC summary. This will do the same as what this one was, this one, right, where we are doing calculating this one, and we are in the calculate QC summary. This is what this Python function will do. See, same only I just took it like if it is empty ribo and hb, then pp sc.pp calculate QC matrix, and finally it will give me this matrix, and it will give me some in JSON format, something it will give, and finally it will read the QC summary nicely. Okay, so this will give me how many of this QC matrix it will give in a matrix format. So now again I have, once QC summary is calculated, I have written another QC function that will just return me this QC summary. Okay, so return the stored QC summary without recalculating. Otherwise, every time it will recalculate, right? So then this function will do the while and plot. Same, see, it is the plot here, but here I am just saving it in a file QC underscore while in plot.png. Okay, now we have the scatter plot. Here user can give X and Y. Okay, so here I have given the instruction that, hey, generate a scatter plot for 2 QC metrics and save as a PNG. Default are total counts and percentage mitochondrial, and user can also pass other QC metrics from the QC summary object. So this gives an agent to instruction that, see, although this function is for two variable, but there is possible, so agent can further ask questions while interacting, like, hey, this is the plot from between total count and percentage mitochondrial, do you want to use some other combinations as well? And you can say that, hey, I want between total counts versus, let's say, ribosomal, and it will again redo it. So it's based on the chatting with the user, it will be able to do. So that's why you have to write your Python function such that it should be more generic, and it should be more modular as well. Now the same thing, this is the for filtering. So I have now comes the QC is done. So then comes the filtering part. So this two filtering, filter by gene and filter by cells. So two Python function, pre-processing filter cells and pre-processing filter genes. Okay. Now the doublet one. Okay. Doublet detection, it's called scrublet one is there. So you see, just make a very simple Python function here and scrublet, and if that is not found, then it will give me a response that scrublet did not add to this server, this object. And finally, if something happens, so it will remove the doublet, and it will say that doublet detection complete, and removed how many cells and how many cells are remaining. Okay. Then once your doublet is done, then comes the normalization part. So these three is packaged into this function, save. Okay, there is nothing different there. Only thing, like, we are just check some, we are using the global A data and doing some check that if it is not present, it will say that that data is not loaded. So now after normalization, we are doing the feature selection. So here I am calculating highly variable genes, and that I have packaged it over here. See, pp dot highly variable genes. Okay. So again, it will give me a response that feature selection is complete. Top this many genes are annotated. Okay. So now we have some plot, right? So these two plots, it will plot it. So that is there in this function is package QC plot highly variable genes. Okay. So now we have the dimensional reduction. Here we are doing the PCA and we are doing some plot as well, right? So that's what this will do, the PCA. That's where I have put that single command of run PCA over here, this one, and QC plot PCA will will plot this one, this plot. And here we have, see here the because we are using this 4 types of plot, right? So here I have explicitly stated that what color variable should be used, so by default it will be color coded by empty, but user can pass other parameters as well. So here we are doing a PCA, and we are giving the color variable with respect to this, but the dimension is like for between first principal, second principal component, 2 to 3, 3 to 4, and 4 to 5. So that way you can see how within each principal component, how clustering looks like, and you can give the color option, choose a color option like that. And then comes the UMAP part. So that's where here the UMAP is in just one line code, and that is what packaged over here. Calculate the neighbors and calculate the UMAP. Okay, and then plot UMAP, the plotting one, this one is packaged in this function plot UMAP, and then we go to the actual clustering. That's where your laid-in clustering, that part is packaged in this function. Okay, run laid-in clustering, and then you are doing the actual UMAP plot of this cluster. Okay, so then you do the plot laid-in clustering. Okay, so this is how you can see that all the functions of the Scanpy, you can define a very, very simple Python function and make it more generic and decorate with this MCP tool, and you can package a MCP server. And if you look carefully, what I have done here to organize this tool, right? This is the most important part, because when you do so many tools expose, right, so your number of tools might increase. So that's where I have prefixed all the tools. So for example, anything pre-processing, that function will start pre-processing underscore, pre-processing underscore, QC underscore, QC underscore valiant plot, those kind of things. So that way, I can later on, when I can build an agentic system where I can have a dedicated QC agent, I can have a dedicated pre-processing agent. So instead of fetching from the MCP server all 25 tools, QC agent will just ask for, hey, give me only the QC tools, or give me only the pre-processing tools. So that way, number of tool response what we get from MCP server will reduce, and that way we will save some tokens. Okay. but you get an idea that like how simple it is to convert the simple Python functions and decorate it. And last at the end, right, I have added some health check here. This is a boilerplate code you can get from fast MCP server. This is just to because when if you want to connect to this server, and you can that URL what we get, if you just type health, it will say all okay. So that means we know that okay, my server is running. Okay. So this is what the code. Sorry for the lengthy explanation, but because the Scanpy itself is very long. So let's come back here. This is my single cell MCP server here, and within that, let me zoom it a bit. Within that SRC folder, and for now you forget about all the other files. Okay, all the files, because that is needed when we actually do the packaging, right? Sharing with the other people. So, but for now, our main code is over here, and so let's start the MCP server here. So I will just move to src single cell. Okay, so this is where my Python function is there. Okay, so how can we run it? First MCP run single cell MCP, then MCP, and just a second. I'm just checking, the code. Okay, this is after that, right? Okay, just a second. So now what we will do, like, minus T is what streamable HTTP, and this is the host, and I'm giving, let's say, which port is running. It is 8083 is running, right? So let's go with 8082. Okay, so with that, it will start the single cell MCP server. Okay. Yeah, let me pause for a minute. Okay, so it started the MCP server. You see, it started in 8082 port, and you can also see in the ports that 8082 is running. Okay, so let's kill the older one. Okay, the NGSQC one. Okay, so this is the my server is running here. So 8082. So I see that now I have only one port is running. Okay, so let's go to this one, and I'm just creating a new chart. Now let's go to files and settings. We'll now configure our cloud desktop with the MCP server. So I can, so this is running, it's saying I go to edit config. Okay, so let me just copy this one. Either I can modify this one 8082 here. Okay, let's copy. Let's do one thing. No, don't save this one, and delete it. Okay, so now you can do an edit config so that it will freshly you can write it. Okay, and I paste this one. So here our port number is what, 80. It's running on 8082, right? So 8082 MCP, and here let's call this, instead of NGSQC, it's a single cell MCP. Okay, so this is the MCP server. Okay, so now we can close this one and go to cloud desktop, go to developer mode, and reload the MCP configuration. So now you see my MCP server is initializing. Okay, so let's see, and also meanwhile you can go and check the logs in the MCP log file. So yeah, so I'm getting some information here, so it's good, there is no error. So now open a new chart. Can you check again, what local MCP server you have? So now it's, see, I have created a grid function. It is trying to call that one, so it called my grid function. So it says now I have access to single cell MCP server, I can, what it can do, it can do data loading, it can do the quality control, it can do the pre-processing, and it can do the dimension detection and clustering as well. Okay, so now you see it load all the capability. So now we will do the data loading part. Okay, so let me see where I have my data is available or not. So I have copied the data right now. Okay, so it is there in my folder here. Okay, so I just take the file name. So this is in which format? H580, the binary format. So this is the path of that server. Okay, so now let's say that, hey, load, okay, data. Okay, this is my first command. So I will load that single cell data for you. So now that's why it is calling the this function, and it gave the path over here. So just allow once. So parallelly, let's check my function as well so that you get idea. So it called this function. Ideally, we should get some response about this one, loaded something. So it will say that, hey, great, the data has been loaded successfully. This appears to be NPBM3 dataset, which is a commonly used example in the single cell analysis. How many cells? 2700, and this many genes were there. This is raw unprocessed data. Would you like me to get a detailed summary of the data? Then calculate QC summary and start with pre-processing steps. So let's go with one by one. So let's call this one. Okay. So now what it will do, it will use the data summary function. So if I do that, it call this function single cell. So it now calling this single cell data summary function. So now it will give me the response in this format. Okay, so now you see it gave in response that this many cells, this many genes were there. These are the 5 barcode, or these are the 5 gene names. So because that's what see, I have enough top 5 I returned. You see, so now it is asking me data is now in the raw format. Would you like to process with quality control analysis? So right, so yes, let's do yes. So let's do yes QC analysis. Okay. So now it will do the calculate QC summary function, right? So then it says that I will run a comprehensive QC analysis. So let me allow that. So now what it will do, it will do this addition of this mitochondria, riboflavin, hemoglobin, and finally it will do this call this function, and it will give me this matrix, the QC summary object it will give me. Okay, so now see it generated, the response is looks like this. Okay, the response what we got, this one. So now it also tried to summarize it in a very nice way. So what is say that, hey, total counts UMI per cell where ranges from 981 to 4903. So in that range, the number of molecules were there, and number of genes detected were in this range. Number of mitochondria percentage range. This was, see, this it used its own brain and it gave some summary to it. So now it asks like, would you like me to generate the Valin plot? So let's go with one. Okay. Third option he gave the both visual. So let's go with 3. Okay. So and also it says that total counts versus this one mitochondrial percentage. So let's go with 3. So now let's go with, so it will calculate, this was done. So now it calculate the QC Valin plot it will do. So you see it used to QC Valin plot. So let's allow once. So now this will actually run the Valin plot, and then it will also asking me to scatter plot. So basically it run this function. So here you see I have generated this the file image will be saved here, and here the image will be saved with X versus Y. So ideally it is running here, right? The server is running here. So he's saying I have generated this file, presented files. Where is the file? So it will try to search where it is. Okay, it struggled that it seems that MCP server saved the plot, but they are not accessible in the current file system. Okay, so let's ask him like, can you generate the plots in you can give this path, data folder. I think I might have done some mistake in the code here, saying that where it should generate, right, the plot. So probably that might be have the mistake here, because I could not I did not pass the So now something, see, now it is using his own brain. Okay. So this is not the way it worked actually. So let's stop this one. So plot thing, I'll have to figure out how it's working. But okay, so we'll say that, proceed with the, start with the pre-processing step. Okay, so only the plot thing only did not work because actually it generated, but somehow it did not, save where. Okay, so now I have asked it to pre-process the this one, so it is calculating that. So excellent. So it's saying that how many cells were. So we get a response that only 0 cells removed. Okay, but now filter genes. So we have filtered this many genes. So what response we got? So filtered this many genes and only 13,000 genes are now remaining. So now it is doing doublet calculation detection. I think it may not work. Okay, it worked because it needs some packages. SK image packages might be needed. So it says that 32 doublets removed. So now we have 2668 cells remaining. So now we can go for normalization. Okay. So here every time I'm allowing. Otherwise, once you know that this is MCP server you have developed, you can see allow also automatically. It will do that. So now what it did, like normalization and log transformation complete. So now it will calculating the total highly number of genes. Okay. There seems to be issue with this function. Let me try with something happened here. Oh, okay. Some Python function might be. Actually, I did try with only once, but yeah, it worked second time. I think first time maybe it did not call that function properly, so it auto corrected and tried. So now it says that 2000 genes are there. So in summary it gives like cell filtering. It did cell filtering, no cells, all QC passed, genes removed, then doublet removed, and final data normalized, and top 2000 genes are detected. So now since visualization is not working somehow, I will see towards the end. But now we can go for run PCA. Okay, so we'll ask them to do PCA. So now it is running PCA and it is calculating number of component around 50. So let me try with the default parameters, but some error came, I think. So let's see. So perfect PCA has been completed with 50 principal component. Let's visualize this result. It will try to do that. It will generate the plot, but I don't know where it is, So PCA plots have been generated, it is saying. So now we can do, run UMap and then perform later. So you can go for, one or both, so we can go for ask him to do both. Okay, we need clustering, right? So now it is calling the run UMap function. It is now doing the UMap analysis. So now it is trying to visualize it, but let's see where does it stores the function. Now it is doing the final clustering, needs to be installed on the, so okay, so I think there is a dependency on this one. So let's go and check what do we need for laden clustering. I think we might need some tools probably. Okay, but yeah, so let me just pause for a minute and find out whether some images it generated were, then I'll get back. Okay. Hey, actually it generated the plot since I was running my server from my server code was here, right? So it generated all the plots there. Okay, so alternately, I could later on improve the codes and explicitly telling where the output directory, but for now you see all the plots it has generated in the folder where my script is there in the src folder. So let's just quickly check you may plot. Okay, and what else plot? So this is what the pca plot it generated, 1 2 2 3 like that. These are the different plots of QC analysis and all. But yeah, so I thought like it did not generate, but yeah, it did generate, and it generated in the where the server is running, because this is running locally, right? And this is where it generated. Okay, and so yeah, I think this some it needed this one. I did not install it, but yeah. So in short, the idea is not to teach you on single cell this thing. The main thing to show you how you can see that how intelligently cloud desktop helps to do the single cell qc analysis. So now I can ask him again that, hey, can you now go with running the pca with, let's say, 30 principal component. I can ask again, can you please remove the filter number of genes like with this criteria. Can you do the doublet connection with this one filtering. Can you do again re-normalize. So that means it's like someone is there for you who is an expert in single cell data analysis, and you are just telling him how to do it. So we can say that, hey, do that, generate the plot, and you look the plot, and if you're not satisfied, you ask him to do again. So that's why it is more of AI assisted research. It will help you to do that. So that was the single cell. So just I will quickly summarize what we did. We did nothing. We wrote simply from the Scanpy tutorial, whatever the functions are there, we just converted that into Python functions, and simply created an MCP server, and just you added only MCP tool, MCP tool, MCP tool. That's all. And then we ran it, and we hosted it, and we configured our cloud desktop to connect to that MCP server. And then we let the cloud desktop, or the AI assistant to assist us for doing this single cell data analysis. Okay, so the idea was to show you like even how complex design. So that's what design I was telling, that you can write your functions such that all qc function you starts with qc underscore name, so that you can apply some filtering to the tool list. Okay, so that's about this video. So where now we built our MCP server, and we did single cell data analysis. So the last is like we need to learn how you can share it with the and other people. Okay, so that's where we will take our code and we will host into GitHub, and so that other people can do like this. They can pull our code and we can do that. So I will just walk you through that into the next video. So yeah, I hope this video was useful. I really liked creating this particular use case, and I hope this was useful. And if you have any queries, anything, please put in the comments. I will try to respond. Also, I will put the code details, everything in the description as well. And yeah, please reach out to me. Thank you so much for watching. Thank you. Bye.